Take it away.
I'm moderating this, so I'd just like to also introduce Ryan, because although I've read his work, he comes to us via, the typical great places, Harvard, Stanford, back to Harvard, UCSD, to Salk, and UCSD, where he's a professor of cognitive science, and probably a lot of other things other departments probably give him appointments to. But please take it away, Ron.
Well, thank you. Thanks for the nice introduction, and thanks, Owen and Howard, for the invitation. I'm excited to be part of Scorch and to see some familiar faces. Owen and I worked closely together as part of the BICCN, which I'm hoping to talk about, which I'll tell you about a little bit today. Let's see. So can you see my slides, I hope?
Yes, we can.
Great. Okay, and you see the presenter view, right?
That's the full view.
Great. Thanks. So yeah, so I'm Aran Mukamel. I'm in San Diego, and so I'm here as part of the UCSD U01, led by Tariq Rana in pediatrics here at UCSD, and with Kyle Golton and also Alan Wong, who's not pictured here. And the title of our project is Revealing the Single Cell Determinants of Brain Relevant to Persistent HIV Infection and Opioid Use Disorder. And we're really working with 10x multi-omic data and other assays, as Owen described, but we're still very much at the beginning of stages of those projects. And so what I'd like to focus on today is to tell you a little bit about the experience that I was privileged to be part of as part of BICCN and some other single cell projects studying brain cells, and hopefully to focus on maybe some lessons that we can learn about collaborating together as a consortium, and some of the challenges and opportunities that come up when we are able to coordinate these really large scale rich studies using these really incredible techniques that really present opportunities for analysis that go beyond what can be done in one lab. So, the thing, just to start at high level, the thing that really gets me up in the morning about science is really trying to think about understanding diversity at the level of both within a brain and across brains. So, we know, for example, that siblings share a common genetic heritage, but that there are differences between us. And I see my graphic is not coming up, so I'll talk about it in a second. And really diversity and variability, both due to experience, drug use, disease status, but other factors are really essential to our humanity. And in my lab, we've been interested in kind of investigating this across dimensions of age and sex and genetic background and et cetera. At the same time, we know that there's an incredible diversity of brain cells. The parts list of the brain includes, we already saw some pictures from Ramoni Cajal. So in terms of the morphology, they're very diverse. And of course, the connectivity, physiology, and gene expression are also quite diverse. And one of the things I've been very interested in is the role of DNA methylation in this diversity. So this is a picture I really like it comes from Siddhartha Mukherjee's book. And it's a photo of his mother and her, monozygotic twin sister. So these are identical twins. So, clearly they share 100% of their genetic heritage in terms of sequence, but they're not epigenetically identical. And in fact, his aunt, his mother's twin sister, is schizophrenic, and his mother is not. And so, he writes about the implications of that for our thinking. So I wanted to start with a little bit of data from a study that's not a single cell study. Here, let me just move my presenter tools. But then I think motivates some of the kinds of questions that we're all interested in. So this is work that we did together with Stella Dracheva at Mount Sinai, and her lab developed techniques for being able to purify different populations of neurons from the human brain. So using antibodies and fluorescence activated nuclei sorting. So we could look at excitatory neurons, inhibitory neurons, and oligodendrocytes. And the type of data, RNA sequencing, DNA methylation profiling, both CG methylation, non-CG methylation, really reveals the incredible richness and diversity of differences between cell types. So you can see here three marker gene loci where you have cell type specific expression and corresponding differences in the specific sites that are methylated or unmethylated in each cell type, as well as other histone modifications, hydroxymethylation. and this is the type of data that for me as a neural data scientist, I think of myself as somebody who's, a neural data scientist, really presents a huge opportunity for understanding the parts list of the brain. One of the applications of this kind of data that I think is quite interesting and exciting is trying to link cell types with specific diseases. So for example, we know that the heritability for a complex neuropsychiatric illness like schizophrenia is widely distributed across the genome. This is a Manhattan plot showing the 108 or so loci that as of 2014 were associated. By now we have maybe twice that number. But one of the challenges with these type of data, of course, is that these types of associations don't pinpoint the exact causal variants or genes that are involved. And in fact, because of linkage disequilibrium, we have a broad region, obviously, that may be correlated with the phenotype. And so using cell type specific information such as DNA methylation, we could try to pinpoint which cell types might have active regulatory elements within one of these loci, and then look for enrichment of these kinds of things. And this is really, I'm showing this just as an example, but we could find enrichment in this case of GABAergic, active GABAergic enhancer, putative enhancer elements in these risk loci for schizophrenia. And then we could look across other cell types and other diseases and make these comparisons. So for example, for a range of neuropsychiatric or neurocognitive diseases, we find enrichments in neurons, and in some cases, both glutamatergic and GABAergic neuron types, whereas control, equally powered GWAS for other diseases had fewer associations. So, but of course, the study that I just showed involved purified neuronal populations, but those are still extremely diverse. And one of the goals of in fact, the first goal of the NIH Brain Initiative, in their vision document was to define the cell types of the brain as a starting point for intellectual and technical progress. And so the idea there is, as opposed to artificial neural networks where we have kind of these anonymous, uniform units that are doing computations, we want a detailed characterization of the parts that make up neural circuits. And one of the reasons is that this gives us a framework for understanding what the cell types are doing, but it also provides tools which can be a foundation for further experiments. So for example, reagents and tools for targeting specific cell types. And one of the things that I think was really interesting that came out of the BICCN, I wonder if Owen and others would agree, is that, over and over we kept returning to the question of what is a cell type and where do we draw the boundaries between cell types? And so there are these conceptual questions that I think are really interesting, but also they don't have a single answer. And I'll say a little bit about some of the approaches that we took to trying to address that. And I think that's relevant for us. In this group, we'll be, looking at disease impact, at the differences, changes due to opioid use and HIV. But it's important to have a common set, a common terminology and a common framework for talking about, how do we know that we're talking about the same cell type across labs, but also across species, across conditions, et cetera. Okay, so the technology that I think is the basis of most of the studies here is really looking at single cells. And this is I think a metaphor that comes, I believe originally I've heard it ascribed to Aviv Regev, but it's really a great way of thinking about it, that traditional molecular techniques involve looking at a smoothie where we're mashing up all of the molecules in a piece of tissue or of brain tissue. Whereas single cell techniques really allow us to look at the fruit salad and really understand the unique and distinctive features of individual cells, and then of course how those individual components are affected by conditions like opioid use and HIV. And one of the really powerful features of single cell analysis is that there's a wide diversity of modalities, of data modalities that we can actually measure in single cells. So here are a few described. So for example, we can measure the transcriptome by collecting cytoplasmic RNA or nuclear RNA. And these have distinct interpretations and advantages. As I mentioned, we've been very interested in DNA methylation, so modifications of the DNA itself. There are also now single cell methods for analyzing histone modifications in single cells, chromatin accessibility, as well as the 3D conformation of chromatin. And in addition, there are ways of trying to pair some of these data modalities with, for example, physiology, such as patch-seq, as well as connectivity, for example, bar-seq and other techniques for mapping the connections between cell types in the context of their molecular identity. And one of the things that I think is useful is to think about these different approaches in terms of the axes on which they're complementary with each other. So, you know, I like to think about them as they're high throughput and fine grained. And so in terms of the throughput, we can think about how many cells can be profiled. And of course, this is partly a matter of cost. So we have examples of really high throughput techniques like 10x sequencing, which I think our group is using, and I think a lot of others. And the real superpower of that technique is that we can get hundreds of thousands or even millions of cells from an individual sample. On the other hand, the y-axis here is showing the number of unique molecules sequenced per cell. And so that can be fairly limited with a high throughput technique like 10x. By contrast, things like DNA methylation sequencing or full-length RNA sequencing with SmartSeq provide much more information per cell, but are more costly, and so we profile fewer cells. Let me skip this. And so again, the BICCN was really it was really a meeting of the minds of many scientists. And with the shared goal of coordinating the collection and production of data, as well as the analysis across several species, but really across data modalities. And so in the first phase of BICCN, and I'll mainly focus on data from the studies of mouse, you know, we had transcriptomic data from many platforms and technologies. We had open chromatin data, DNA methylation data. And so here's an overview of the type of the scope of those data sets. Again, they varied by orders of magnitude in terms of how many cells were profiled, but as well as in terms of the depth of sequencing per cell. So this is similar to that plot I showed you earlier, but now each of these dots is one of the 9 data sets that contributed to the BICCN. This was the first phase of the BICCN, where all of the groups coordinated studies of the mouse primary motor cortex. Currently, the groups are working on, you know, doing a similar effort to characterize the rest of the mouse brain. But in terms of the primary motor cortex, we had these 9 data sets. And again, some of them had very high throughput, but a relatively more modest number of data points per cell, and vice versa. You know, we had these transcriptomic and DNA methylation data sets that were tens of thousands of cells, but millions of molecules per cell. So what can we do with those kinds of data? Well, one of the goals of the BICCN, which I think, you know, the idea was to set the stage for the kind of work that we're going to be doing here in Scorch, as well as in other studies, for example, the Cycon code that I'm also involved with. One of the goals is to really characterize how can we define cell types, and that really involves understanding the impact of different dimensions of the parameter space on which we can characterize cells. So for example, how does the number of cells that we actually are able to collect affect our clustering and our analysis of the cell types? How does it depend on the depth, the number of reads or the number of genes that we profile per cell? How does it depend on the particular molecular features that we're measuring, such as gene expression versus the epigenome? We want to define cells in a way that is hopefully concordant between these different modalities. It can also depend on the particular assay, the technology. So even if 10x RNA-seq and SmartSeq are measuring the same molecules, they still have technical differences, which could lead to either differences or consistencies in the cell types. We talked a lot about data processing. This is something that Owen and others were very involved with in trying to coordinate our data processing to ensure high quality, reproducible, and convergent results across the labs and techniques. As well as, you know, the spatial location of cells, their physiological features. We didn't necessarily solve all of these problems, but this was kind of the landscape of questions that we were interested in trying to address. and as well as cross-species validation. So there was some, attempts to map the same cell types across species and to understand how conserved or variable were they. So let me talk specifically a little bit about what we found in the mouse motor cortex. So this is data, the transcriptomic data from about 500,000 cells from the mouse primary motor cortex. And again, they were measured by, this data was produced by primarily by teams at the Allen Institute, as well as the Broad Institute, led by Evan McCosco and Honghui Zhang. And so these data sets, these cells are colored either by the specific data modality that they came from or by the assigned cell type. And what you can see, first of all, is that we are able to, for the most part, overcome the really substantial differences in the between these modalities, these data modalities, as well as they're produced by different labs from different mice, and cluster them together and identify, for example, layer 5 intratelencephalic neurons that have a consistent signature across these data modalities. Of course, there are also gradients. So here you can see that within a cell type, there's a gradient, for example, between the single cell RNA versus single nuclei RNA measurements, which is interesting, and that's something that we could characterize further. In addition, we could do the same kind of analysis separately for DNA methylation data. This is about 10,000 DNA methylation profiled cells, and another set of, I believe it was something like 80,000, 80,000 open chromatin measured cells. But a priori, we didn't have a way of combining these because these are not multi-omic measurements. Now we're starting to do more increasingly multi-omic measurements. But even with multi-omic measurements, we have the challenge of how do we merge and integrate and fuse these types of data sets. So that's something that my lab was very interested in, and what we decided to think about this as a missing data problem. So the idea, one way of thinking about it is that for each cell, we have a certain set of features that are measured. So for example, for a cell that was measured by a single cell RNA-seq, we've measured the gene expression from tens of thousands of genes, but we're missing information about what the DNA methylation pattern was and what the chromatin accessibility pattern was. And vice versa, for cells that we measured by methylation sequencing, we've observed their methylation pattern, but we're missing information about the other two modalities. So the question was, how can we link them? And we took advantage of the fact that these data modalities are distinct. They're giving us distinct information, but at the same time, there's very strong correlations at certain features. So for example, here, this is the locus of the gene TAC1, which is expressed in a subset of parvalbumin expressing GABAergic neurons. And what you can see is a high level of gene expression in two different data sets here in purple and red, as well as open chromatin, so a TAC-seq density here throughout the gene body and at the promoter especially, as well as low DNA methylation. So there's this correlation within the gene bodies that we could use by correlating across all of the gene bodies, we could try to link these data sets. So the idea is we've measured, again, separately each of the modalities in separate groups of cells, and what we'd like to do is integrate them together. And again, hopefully this is the kind of thing that we'll be able to do, I'm hoping in Scorch as well, to link across the different data sets that are being produced. By now, there are actually multiple algorithms and procedures for doing this, and what's been striking to me is how successful you can be at doing this linking with many different approaches. I'll tell you about one computational method that we developed, but I think the reason it really succeeds is because these different epigenomic and transcriptomic signatures are very consistent among each other. So the approach that we proposed is to look for nearest neighbors. So the idea is we take each cell in one data modality, for example, this green cell, and find a set of maybe 10 or 20 most similar cells in each of the other data sets. And we do this in a reciprocal fashion. So again, for each of these purple methylation cells, we find its nearest neighbors in the corresponding transcriptomic data set. And once we have those neighbors, we can average their, for example, methylation pattern across these purple cells, and then fill in the missing data about what the methylation pattern was for these cells. Once we have this kind of imputed data set, we can then do a joint analysis with a low dimensional embedding and a clustering that uses all of the cells. So this is a visualization of what the data looked like from 6 different data sets. Again, some of them we have lots of cells, 130,000 cells from 10x, fewer cells from methylation, but of course the methylation gives us important information that we want to integrate into the context of these cells. And so we imputed the data, and this is just showing some of the results. You can see now we have all 6 data sets embedded in the same space, and each of these plots is colored by the clustering that was assigned separately by each of the data analysis groups. The data production labs did separate clustering of their cells, and what you can see is that there's a lot of consistency between them. We can put them together and again see that they're highly consistent. I'll mention that we took another approach as well. So Josh Welch used an algorithm he developed in Evan McCosko's lab called Liger and achieved similar kind of successful integration results. Once we have these kind of consistent embeddings and annotations, what we can do is look at the expression level and the epigenetic marks at specific loci. So for example, here, this gene TSHC2 has a specific pattern of very, very cell type specific expression in deep layer excitatory neurons, as well as open chromatin and DNA methylation in the very same clusters of cells. These are not the same cells because the experiments were done separately. But we can also look across other marker genes. And then importantly, we can look at the data in other ways as well. We can look at single base resolution here at tracks showing the DNA methylation, the attack-seq, and the RNA across all of the, in this case, the glutamatergic cell types. So one thing that was really important in the BICCN, again, was talking a lot with each other about how we could coordinate the data for organizing, sharing, and collaborating, together on these questions. And this is still an ongoing effort, but with folks like Owen and his team at NEMO, Tim Tickle at the Broad Institute, Mike Harlowich at the Allen Institute, Marianne Martone and others, there was really a big effort put into organizing the data for access. So here you can see a screenshot of the NEMO portal for searching and being able to download the data from each of the studies, and also organizing the data from the different grants. And I think this is a very it's really important, I think, for a collaboration like this, for folks to know where the data is, to know what, to have sufficient access to metadata and descriptions of samples that they are able to have a sense of what's available and what can be brought in to analyze in the context of their study. So there were many different efforts at building tools for visualization and analysis of the data. Here's a list of some of them. This is a screenshot from something that we developed in my lab for being able to scroll through some of the epigenetic data that was produced. And then in each of those data sets, one could create these two-dimensional embeddings and color them by the DNA methylation at specific genes. Here's some more screenshots of that. You could also toggle different parameters of the analysis and then look at the data also in terms of violin plots and that kind of thing. So I think the good news is that there are now many platforms, software platforms and other tools that can be brought in for this. But I think one thing that's really important is that just having the software is not enough. I mean, there's a lot of work that needs to go into the organization and the really thoughtful organization of the data in a way that allows users both within the consortium and outside to understand and appreciate the scope of the data and be able to use it. Here's another screenshot of that browser that I mentioned. So this is a way that users can, and maybe I'll take the risk of doing a live demo and just click on it.
Yeah, we're at 5 minutes left.
Okay, great, thank you. So this is a live demo of this browser, and it really allows users to, for example, select which cell types they want to show. For example, I can bring up the can show all cell types, select which data modalities. I can add the RNA-seq data, for example, and reload it. And then users can also then search for specific genes to look for cell type differences. So, in the context of Scorch, I think, I think it would be really exciting to be able to have this kind of data set from the human brain where we're actually able to see, for example, differences across conditions and not just across cell types. So users can then type in a gene, for example, TSHC2 that I mentioned earlier, and go to that locus. Maybe I won't wait for that to come up. Okay, so in the 5 minutes that I have left, I'd like to just mention a couple of the kind of analyses that we wanted to do with these data as part of BACCN. And I think a couple the speakers this morning, I believe John might have mentioned that we're really interested in making the data reproducible and rigorous, in the sense that we're investing a lot of time and resources in producing these data. And so we want them to be reusable, and that means really thinking about characterizing in as quantitative a way as we can the reproducibility of the annotations and the cell types that we define, and also of then the downstream effects. So one approach developed by Jesse Gillis is an algorithm they call MetaNeighbor, that essentially looks for nearest neighbors between cells across data sets, similar to what I mentioned earlier, but then uses those nearest neighbor graphs to compute scores for the cluster annotations, for the cell type annotations, that really tell you about how well the cell type annotations in one data set are supported by the data in a separate data set. So for example, if we can make a plot like this that shows the score, the reproducibility score, across every pair of clusters that were generated across each of the data sets. And so these diagonal blocks show you the high level of reproducibility for example, for CGE-derived GABAergic cells across all of the data modalities. We can zoom in here to just the MGE-derived neurons. I apologize for the pixelated quality here, but what you can see is that even the subtypes of MGE-derived GABAergic neurons have a high level of consistency across data sets compared to their similarity to other cell types even in the same data set. Let me skip this for now. And another question that we wanted to address was this question of lumpers versus splitters. And this gets to what I mentioned earlier. Two researchers looking at even the same data set can come to very different conclusions about where the boundary between cell types lies. And this this was the idea of lumpers versus splitters goes back to Darwin and probably earlier, when he was thinking about categorizing species. But of course, the same thing comes up when categorizing cell types. So the kind of things that that we have to think about are how fine-grained do we want to go? We can divide our cells into very fine-grained cell types, and that has certain advantages. But from the perspective of a lumper, and I have to say, so my background is originally in physics, and I think physicists tend to be more lumpers, and biologists often are more on the splitter end of this spectrum. We want to know, are these fine-grained distinctions that we're defining biologically meaningful? Are they statistically reproducible? Can we really observe them across data types, for example, in both the epigenome and the transcriptome?
For 1 minute.
Okay, thanks. So I won't get into the details, but the method that we came up with for trying to look at this involved, again, comparing across data sets and across analyses. So here, just schematically, you can imagine taking the same data set and analyzing it at a very coarse-grained level or at a very fine-grained level, and then trying to use a kind of cross-validation to split, for example, the genes as well as the cells in a way that allows you to assign clusters based on half the data, and then test whether those clusters are consistent across data sets using a separate held-out data set. And so let me not talk about the details of it, but just show you we could analyze the same data set at a number of resolutions, and get anywhere from 10 to 1,000 clusters. So that's just a choice that a researcher can choose to make based on a parameter in the analysis. And if we look at the a measure of the error that we get in the training data. So in other words, in the cells that were used for the clustering itself, well, the more fine-grained you go, the better. The cells are more accurately described. But of course, that's not really what we care about. What we care about is this kind of test set error where we're asking how well are we describing held out cells that weren't used for the clustering. And so you can see there's kind of a sweet spot in the center where roughly in this case, something like 50 to 100 cell types were well supported by this particular data set.
And final
So
thought.
I think I'm out of time. I'll just put up my conclusion slide. And just say, I think there's really a bright outlook for this kind of analysis across many different application areas. I think it's really exciting in this setting that we're going to start to use these data to look at disease and opioid use disorder. I'd really urge people to think about how can we take advantage of the complementary, strengths of the different data modalities. And yeah, let me just put up the collaborators, and I'm happy to take any questions if we have time.
Yep. So up next, we have a 15-minute break. So
great.
Depending on where you're at, we'll be meeting at 1:30. So at 1:30 for our Eastern folks and 10:30 for our Pacific and everywhere in between. And so we do have time if you want to answer a question or two.
There
Let's
is
see.
one question in the Q&A.
Okay.
That is,
how can
let me read it. How can we access reliable and acceptable mathematical models for the analysis? Looks like algorithms are key for reliability and comparable analyses.
Yeah, that's a great.
Yeah.
Thank you. Yeah, that's a great question. I mean, I absolutely agree that I think algorithms are key. I think it's important to keep in mind, first of all, that this is a space where the algorithms, just like the data, the data production technologies are rapidly progressing. I think the algorithms are also making a lot of progress. As I mentioned, I think the good news is that there's a lot of consistency between different computational approaches that people have come up with for doing the clustering and for comparing and doing data integration. But at the same time, it can be quite, I think, overwhelming, the flexibility, the number of different choices that are available for doing these analyses, especially because some of the questions, such as, what is a cell type, those questions don't necessarily have a single ground truth answer. So, I think one lesson that I take away is that there's not going to be a single gold standard, and that we need to get used to thinking about doing multiple analyses of the data, and really, then thinking about what do we learn from looking at the data at different levels of resolution. But I'd love to hear if others have thoughts on that as well.
I certainly agree. And, the trick, as with other things, is it's not just that 3 of the 5 showed you one answer and therefore that's correct, right? So
yeah, we want findings that generalize. And so,
right.
An advantage of a consortium is you can look at the same question across different data sets, different modalities, and if we're finding robust phenomena, then they should be reflected across the different studies.
Thank you. And I think we'll give people a little break now.
Okay, great. Thank you so much.
So Joe, we come back at 1:30. Okay, thanks.